Good afternoon. Thanks for the opportunity to be here. I'm going to provide a regulatory landscape update on biomarkers in cancer immunotherapy. So here's my outline. I'll provide what are the current immuno-oncology biomarkers that are FDA approved. We'll touch base on companion and complementary diagnostics. We'll talk about the challenges still we are facing with some of these biomarkers. We'll also touch base on tissue-agnostic drug development, and lastly on ctDNA for immuno-oncology-based therapies. So I think we heard a little bit earlier, what are the biomarkers that are currently being used for selecting responders to cancer immunotherapy. Of course, first is the PD-L1 and MSI high, MMR, DMMR, and TMB. So I think I'll go over this later, so we'll skip the details of this here. Just want to show you the relationship between these three biomarkers. It was published a couple of years back, but still is really a good slide, I thought, to see how in multiple cancer types, there were 26 cancer types. They had 11,348 subjects, and looking at the relationship, they are, you can see they are related, some of them, but still PD-L1 is big in terms of the prevalence, and all three are unique biomarkers, so each one has its contribution in cancer immunotherapy. We heard the term companion diagnostics, so I'll just go over how we define it. They are the biomarker. They are the tests that are providing information that is essential for the safe and effective use of a corresponding therapeutic product. But at the same time, when we were dealing with PD-L1 and immunotherapy, we also ran into some of these. Even the PD-L1 itself, in some cancer types, they were when there was the all-comer trials, the biomarker. The positives would respond better to the drug, but so there is this aiding in the benefit risk assessments, but they are not really prerequisites for receiving the drug. So that's there is no guidance for complementary diagnostics, but this is the draft definition for complementary diagnostics. But again, the decision whether it's companion or complementary, it's based on the trial design and also depending on the outcome from the clinical trials. And here's the link to all the FDA cleared approved CDx. Another, what I wanted to point out is the indications for the tests are different between companion and complementary. As I said, companion diagnostics, mostly all of them are based on selection claim. That's mainly because only the biomarker positives were studied. And so as you can see how the indication for the test is, it says it's as an aid in identifying the cancer patients for treatment with the drug. Whereas the complementary diagnostics were all-comer trials, and the device IU, it says, may be associated with enhanced whatever the endpoint for that trial. So getting into what are the assays that are approved for PD-L1, there are 4 different antibodies. You can see here, 22C3, 288, those are both from Dako, Agilent, and SP263, SP142 are from Ventana. And the respective cancers are also actually listed here. But there are, as you probably already know about this, I'm probably preaching to the choir here. So this is an example of the 22C3 indications for use. You can see there are different scoring and cutoffs. So for non-small cell lung cancer, it's TPS is the scoring, and the next slide I talk about those. And for the rest of it, it's CPS. And the different cutoffs, you can see there's 1, there's 10. So depending on what was the cutoff that was used for the clinical trial, that's the cutoff that was approved. And for non-small cell lung cancer, there are 2 drugs approved with 2 different cutoffs. So what is TPS? That's when they are scoring only tumor cell staining. And when they're scoring tumor cell and immune cell, it's a combined positive score. That's the CPS. And then immune cell staining only, that's the IC. And that's for, so SP142 has only immune cell staining scoring. So we actually saw what was going to happen and convened a workshop in 2015 itself, to show how this is going to actually create some issues down the line. And there was a blueprint PD-L1 IHC comparability project. There was a multi-stakeholder harmonization effort that was formed. And as you can see from here, SP142 it behaves differently. It stains fewer cells than the other 3 antibodies. This is published, so the references here. And it's still, all these challenges that we discussed, it's still there. The various cutoffs for PD-L1 positive versus negative, for or sometimes high versus slow, and different scoring systems, and depending on the clone, there is differences in scoring as well as differences in the cutoff. So moving on to the 2 other biomarkers, MSI high, DMMR. This DMMR was shown to be really effective for immunotherapy in 2015, the article that's listed here. And later on in 2017, Keytruda was granted an accelerated approval for solid tumors. So the difference between this and all the other approvals where this was, the approval was based on a biomarker and not based on a cancer type. So it is, tumor agnostic drug development. And the companion diagnostics was not contemporaneously approved. It was a post-marketing commitment. And that was in 2017. Then 2021, there was also another approval for Jemperli. So, they also got the tissue agnostic approval for this. And another biomarker we heard earlier about the tumor mutation burden. This is also a really novel biomarker. The 2020, that's when Keytruda was again granted an accelerated approval. So again, for solid tumors, the approval is based on the biomarker and not just on the cancer type. So for this, a companion diagnostics was contemporaneously approved, Foundation 1 CDX, and for that assay, the TMB is measured by counting all synonymous and non-synonymous substitution and indel variants present at 5% allele frequency or greater, and then filtering out any potential germline variants. So, it is actually the mutation number is then divided by the coding region. So that's why it's the mutations per megabase. And for that particular approval, 10 or greater was chosen as the cutoff. Again, we saw some sources of variability in the TMB measurement. So because TMB measures can vary depending on the pre-analytical conditions, it could be due to the analytical factors and also the bioinformatics, what's the different algorithms that could be used for filtering out the germline variants. So to address these variability, Friends of Cancer Research helped with the harmonizing the TMB measurement, and this is also published, and actually Diana is in the audience. So it's still TMB. There we have continuing challenges with this, which we see that because for the tissue TMB, whether 10 is the right cutoff, and should different tumors, because this is approved for all solid tumors, certain tumors should have different cutoff. And then, of course, we heard about circulating DNA, how you can measure the tumor mutational load from using blood. But for the blood TMB, for an assay validation, we look at the accuracy and what should be used for for accuracy, because you can't use the whole exome sequencing that was used for tissue TMB. And also, how do you actually measure the limit of detection? Again, for tissue, we used computational tumor purity, but that's not possible for this. So, there are some challenges, analytically validation challenges of blood TMB. I also want to touch base on, in general, what are the challenges for tumor agnostic drug development. Of course, the logistical challenges, because these are global trials, so from different countries, so many different sites. And we also understand the incidence of biomarker is different between cancer types, and it could be, and the enrollment challenges are, it's so hard to actually get these rare tumors. And if we have to look into pediatric population, do you use the same dose or it's a different dose? And we heard the companion diagnostics could, if you have to have a diagnostic approved at the same time, sometimes it's not possible because of some of the challenges, like for MSI high DMMR, this was already in standard of care. So there were local tests that were used for enrollment. So how do you get the samples in time to make the validation for companion diagnostic? And of course, when for different biomarkers, are you straight going to tumor agnostic development that was for the TMB, or are you going with specific tumor types first, like the DMMR was approved in some cancers first, then went into the tumor agnostic? So which is the right way to do it? So there are some articles that are published from FDA itself that may be useful in these situations. As we heard some other biomarkers in the earlier talks, there are so many promising biomarkers, such as, I think we heard the tumor microenvironment, T-cell inflamed tumor microenvironment, the interferon gamma related biomarkers. I think there are also trials on like the LAG3, TIGIT. So there are promising biomarkers, but I just want to touch base on ctDNA. Because there are so many different trials happening in this area. So this is helpful to guide, at least the trials are trying to see with how it can guide treatment decision making for immunotherapy. It can be also used for the adjuvant. There are different trials in the adjuvant, neoadjuvant settings. And there are different efforts to looking into, can it be used to predict response? And FOCR is also, again, doing another project, CT Monitor project, looking at the lung cancer immune checkpoint inhibitor clinical trials. And we heard there are some biomarkers could be used for mechanism of treatment resistance. So can you switch therapies? Can you use ctDNA for that? Can you use it to differentiate between pseudoprogression versus true progression? But still there are, again, just like other biomarkers, there are challenges for ctDNA assays because there are different methods that are used for MRD, especially for there are tumor-informed methods, tumor-naive methods. Some are using some smaller panel. And so the standardization of protocols probably needed for consistent ctDNA measurement, and also for protocols for measurement of ctDNA and the different scheduled assessments. Again, there are different units used for measurement of ctDNA, so the harmonization is probably needed in that area as well for use of these in future immunotherapy area. With that, I would like to thank everyone who has worked in, and helped me with this field. So Dr. Beaver, Dr. Thoreau, and Dr. Valenki, she has actually published there is an article that is coming out on this ctDNA for immunotherapy, so that is accepted for publications. And I also want to thank my colleagues in CDRH.